Hello, everyone. How are you today? So today we're going to dissect a daffodil. Yes, dissect a daffodil. Now, you can watch this video, but I encourage you and I recommend that you try to find your own daffodil too, and of course, do your own dissection as well. This is the time of year when daffodils are blooming, and we have a number of them in our backyard, so I thought during this time of quarantine, staying at home, and students not at school, I thought I would do a quick dissection of a daffodil here. So you'll see what I have here. I have four different daffodils, and I have one variety here on the left, and we have the same variety. These three are all the same variety here of daffodils, and I tried to get them at different stages of their growth and development. Where we have this guy here on the right, he's a pretty new one. We might even say it's a bud, but it's ready to open. We have one that's a little bit further along here. We have one that's totally open here as well, and then this is the other variety that's completely open. I couldn't find any buds for that one, so sorry. So anyway, I want you to notice that with this one, if I turn these three upwards, you'll see that how you can see the progression of the petals as they begin with the tightly folded up bud on my right, and progressing through to the totally open one here on the left, this guy here. So anyway, let's take a look at these, and we'll cut them open and see what's inside. So you'll need a knife, and I happen to have this one here, and which is kind of cool about this actually. My name is Mr. Tinker, and this knife happens to be a Swiss Army knife, and the name of this model is the Deluxe Tinker. So that's pretty handy. I had to get this knife, of course, because they named it after me. What an honor. So I'll go ahead and I'm going to cut into this one here. This is the one that I'm going to use for the dissection. I'm going to cut into the stem. I'm going to cut it across here horizontally like this, and then I'll cut it vertically this way so that we can see the inside of it, and I'll describe its texture and give you an idea of what's going on here. So if you can see on the inside of this, it's very glistening. It has a glistening surface to it, and the reason why is because when I cut this open and I bend it a little bit, I'm breaking open some of the cells, and as a result there is water that is being released from vascular tubes called xylem, and there is also some food being released from another set of vascular tubes called phloem. So xylem transports water up through the stem, and phloem transports food and nutrients, minerals, sugars, those types of things to nourish the plant. And so I rub that between my fingers, and it's definitely wet. But also at the same time, once the water evaporates, there's a little bit of stickiness to it as well. So that indicates that there's sugar that's coming out of these cells as well, again from the phloem. So that's something interesting, even about the stem itself. On the outside of the stem, notice it's green, obviously, because of the chlorophyll, but at the same time, it has a waxy cuticle on it. And waxes are one of the three predominant groups of lipids. You have fats, oils, and waxes. And many plants possess these waxes to inhibit the loss of water. And also to protect the plant as well, to give it an outer layer of protection and some strength and stability as well, working together with the carbohydrates. That's giving structural integrity to the stems too, to hold up against the wind blowing on the stems, and of course force of gravity as well, and to keep the stems upright. So there's a good deal of that in here, which the daffodil flower needs. Okay, so one thing that I wanted to point out and maybe make a comparison with, the next structure we're going to look at, is what's called the spathe. And that's this layer of tissue here that's dried out, and it's kind of crunchy as well. The spathe is dried out now because it's no longer needed by the flower. If you look at these other two here, you'll see that this one it's a more recently opened flower. the spathe is not quite as dried out. It's more pliable and not quite as brown. And you'll notice that in this one as well. This spathe is still kind of hugging the flower. And there's even a hint of green in it as well. It's in the process of dying. And again, it's no longer needed. But if this were a tightly wound up bud and was very small, this spathe would be covering it and protecting it. Because the bud is delicate and it needs an extra layer of protection around it until the flower is ready to open up. And once that happens, the spathe is no longer needed. The flower is much larger than the spathe. And plus, of course, it's going to open up and it doesn't want to be enclosed by this tissue that surrounds the immature bud. Okay, so again, that's called the spathe. I can go ahead and pull that off of there and you can see it peels off right off the stem. Next item we'll look at are the petals, okay, which is the most obvious feature of the flower would be the petals and the center part as well. What's interesting about the petals is that there are 3 outer ones and there are 3 inner petals. Okay, so we have 6 altogether, and I'll peel them off one at a time. I'm going to peel the outer ones off first. So there's one, and the second outer petal, and the third outer petal. And then we have 3 inner petals then that are left behind here. So 4, 5, and 6. Okay, now these petals, of course, are there for attraction, to make the flower look pretty to us, and to attract pollinators as well. Now, this center section here, the what's called the more commonly called the trumpet of the plant or of the flower, this is actually called the corona. Now, with what's going on right now, you're thinking corona. This is not the same corona. You will not get a disease from this corona. It's not contagious. Corona just means crown, and it's kind of the shape of a crown, I suppose. It's considered to be the crown of the flower. Okay, again, that's called the corona. Go ahead and tear the corona down the sides here so that we can see the internal parts of the flower then as well. And if we rip the corona down, you'll see that there are a number of structures that will be evident now. So there's one half of the corona, and you'll notice the pigmentation in the corona is darker too, by the way, just more for attraction. Now, speaking of attracting the pollinators, the daffodil has been cultivated to, as you can see in this one as well, into hundreds of varieties. And that was all done artificially over the course of the past few centuries, where plant breeders wanted to get different cultivars or varieties, you could say, of these flowers. And when plant breeders do that, it comes at a cost. So it gives up nectar production or its fragrance in order to get a more beautiful looking flower. In terms of, from a human perspective, we find these to be beautiful. Of course, insects, they don't care about what the flower looks like or the shape of its petals. But to us, that's important. And so over the course of the years, plant breeders have made a number of them. So what I'm getting at here is when I was outside getting these, I had no fear of bees. There were no bugs around them because it's kind of chilly outside and it's very windy. And so the insects are not very active. So rather than relying on insect pollination, daffodils will rely on wind to carry the pollen grains. Anyway, I just wanted to point that out about the daffodil, which is a highly hybridized species of flower. But anyway, moving on with the dissection. So we'll see that there are, I'm going to pull apart these smaller guys right here. They're more filamentous. And these are called the stamens. And I'm trying to separate them, and I'm having a bit of a difficult time. There are 6 of them altogether, 6 stamens, which coincidentally is the same number as the number of petals on the flower that surround the corona. And you'll notice that on the stamens they have a tip that looks kind of fuzzy. And that fuzzy tip, all that fuzziness is, can you guess what it is? I'm sure you can. It's pollen. That's right, it's pollen. So this pollen covers the tips of these stamens, and the tips are called the anthers. And pollen grains are all over it, and you can see how I'm getting much of the pollen off onto my fingers. Okay, so pollen grains, those are what would be considered the quote male reproductive cells, and the pollen grains then will contain half the number of chromosomes to fertilize the quote female reproductive cells, which are called ovules, which we'll look at in a minute. So I'm going to tear off these stamens here just to remove them. And by the way, if you remember from your biology class, whenever that may have been, this is what a particular botanist did, geneticist, the most famous one of all, lived in the 19th century. He was a monk. Any guesses? Yes, Gregor Mendel. This is how Mendel prevented self-pollination of his pea plants. He removed the stamens before the pollen had formed on the anthers. So he removed them so that the pea plants would not self-fertilize. He wanted to do cross-pollination. He wanted to pretend like he was a bee. And so he would take pollen from the anthers of another pea plant and deposit them onto this structure right here, which is called the pistil, specifically on the top of it, which is called the stigma. So he took a paintbrush, a very small paintbrush, and would pick up some pollen grains and then dust it onto the tip of the pistil. And then the pollen grains would move down through the pistil. The stalk of it, by the way, is called the style. Of course, there's a name for everything. So overall, this is called the pistil. The top of it is called the stigma, and then the stalk is called the style. So at the very top of the pistil, right here at the very top of it, there's an opening in the middle of the stigma, and that is the opening through which the pollen grains will be deposited and travel down through. Okay, so this is just a hollow tube, and I'm going to go ahead and remove the pistil as well, so that we can then see what's inside this bulbous part of the flower here. This part is actually called the ovary. And inside the ovary are the female reproductive cells. Again, they have half the number of chromosomes. So pollen contains the what's called the N number, or the haploid number of chromosomes. And the ovules contain the haploid number as well. So whenever the pollen cells meet the ovule cells, you now have the haploid meeting with haploid, and now you get the diploid number of chromosomes. Now, I'm going to try to dig these out here with my knife here, which, by the way, is called a Tinker Deluxe, in case you forgot about that. These are the ovules here. These small, almost transparent, translucent little structures here, and they're large enough that you can actually see them. So they will then become fertilized by the pollen grains. So that's about it. This concludes our dissection of a daffodil, and you probably have never considered dissecting a daffodil before. I hope this video has encouraged you to do so, will motivate you to do so, and take care of it soon, because the daffodils, they're not going to be around for much longer. So that's it for today, and thank you for watching.